Good morning, everyone. So today we're going to discuss about DNA polymerase. DNA polymerase is an enzyme which is involved in DNA replication. So why DNA replication is important is because if we have, let's say, one cell and it has to divide into two cells, then before the division of cell we have to make sure that the genetic material is dividing, and in order to divide that genetic material we need something called DNA polymerase, which will be involved in the replication of DNA. In order for DNA polymerase to function, it requires two things. First is DNTP, could be adenine, guanine, thymine, or cytosine, and another thing which it requires is the primer. So it cannot be like that let's say this is the template strand. Okay, so I'll make template strand with the red color. So let's say this is the template strand and it has this base pairs on it, and it cannot happen that this enzyme can just sit here and start replicating. It cannot happen because this enzyme essentially requires a small primer beforehand. So let's say this is the primer. So if this primer is there, a small primer should always be there in order for that DNA, in order for this DNA polymerase enzyme to sit on that strand and then start extending this primer. So this has to be here. So it requires two things. One is the DNTPs and the another one is the primer in order to replicate one strand, and obviously the template strand. Then we'll see like how what is the structure of this enzyme and how this structure and what all parts are there. So as you can see that this enzyme has the finger, thumb, and palm region. So we'll start with the palm region and we'll discuss like what palm region is exactly, why it is there, and what is the function of palm region. So we'll start, let's say this is the enzyme and it has these are the finger, this is the palm region, and this is the thumb region. So here if we have a let's say we have this template here. So this is the template, and here we have the primer also. So this is the primer here. So once you have this template and primer, the palm region, why it is important is because it has two functions. So this one function is that it has Mg2 positive. You might have heard of Mg2 positive. It is involved in, it's a cofactor for the replication. And the another thing it has is that it forms hydrogen bond. It form hydrogen bond with the strand, newly formed strand. So let's say in here this might be the newly formed strand, this here, right here. So this palm region will form the hydrogen bond with this newly formed strand. Why this hydrogen bond is important is we'll discuss next. So first we'll discuss like why Mg2 positive is important, what exactly is the function of Mg2 positive here. You might have heard this is cofactor, but you might not be knowing what exactly is the function of Mg2 positive here. Okay, so here as you can see, so here this particular thing is the palm region, and in that palm region this is the template strand. I'll just highlight this, and this is the primer. This is the 3 dash end and this is the 5 dash end, this is the 3 dash and this is the 5 dash end. So this is the 3 dash end OH, and this one 3 dash end OH, and these two are the positive ions present here. So why exactly we need these positive ions? As you might have figured out until now, you can see that this is the newcomer DNTP. See this whole thing is the newcomer DNTP, and this is the positive ion. Two positive ions are present. So one positive ion is involved in association with the negatively charged O negative in order for it is stabilizing this thing. So once this reaction happen, this bond will break, and once this bond is broken, then the pyrophosphate will form. So the pyrophosphate has to be stable. In order to stabilize this whole negative charge here, as you can see here, this negative charge, this one ion is present. The another ion has a different function. Why it is present is because its association with this oxygen negative charge will lead to the nucleophilic attack on the phosphate, and this nucleophilic attack will lead to the polymerization. We have two ions. One is involved in increasing the negative charge on the oxygen, and another one is involved in stabilizing the pyrophosphate, which will be released after the reaction. So this is the two function of these two ions which are present here. Then the another function of this palm region is the formation of hydrogen bond. So this hydrogen bond is formed, and because of this formation of hydrogen bond, it will only be formed if the complementarity here is correct. If the complementarity is not correct, the hydrogen bond will not form, and if the hydrogen bond will not form, this configuration will change, and because of this change in configuration, the exonuclease function will come up, and how it will come up we'll discuss later. But right now you just remember that this palm region is there, which has two function. One is hydrogen bond formation with this newly formed strand, and the another one is Mg2 positive, which again has two function, which we just discussed here. So the another one is the finger region. So we have studied the palm region. Now the another one is this finger region. So this is the palm and this is the finger region. Again, the finger region will have two functions. So one is the changing the configuration, open and close configuration of the enzyme. So here, let's say this is again I'm making the templates. Let's say this is the template, and this is the primer on the template. This is the DNTP. Once you find the complementary DNTP to this template base pair, then what happen is, because of this complementarity and because of this association, the finger region will come up like that. So it will change its position and it will come up like this. It is like 40-degree, it will bend, and because of this 40-degree bend, what exactly will happen is let me show you. So yeah, so here, so this is the finger region, as you can see, and here this is the bend finger region. So it was like that earlier, and here this is the palm region. As you can see, this is the palm region. So these are the two negative the two ions which are positively charged. So if you can see, this is the incoming DNTP, and when this incoming DNTP is coming, and when you know that this incoming DNTP is fit for this, this is complementary to the base pair in on the template strand, what will happen is that it will come like 40-degree forward, and because of this coming 40-degree forward, this association will happen with the phosphate, which is negatively charged, and this tyrosine will associate with the upcoming new coming base pair. And because of this 40-degree, again another thing which will happen is that these positively charged, they will not move, but because of this movement of this finger region, they will come closer, this phosphate ions and this positively charged ion, and because they are closer now, this reaction can happen. And because of so the reaction is exactly happening because of this closed posture of this enzyme. So once this reaction happens, after this, once the bond is formed, once these hydrogen bonds are formed, and once this this nucleophilic attack happens and pyrophosphate is released from there, then the configuration will go back. So this will again open up, and again it will wait for the another complementary base pair to come up, and then again it will bend 40-degree and form this reaction. So it has to bend again and again in order to polymerize this reaction. So this is the one function of the this is the one function of the finger, the opening and closing. And the another function of finger domain is that it will give a 90-degree turn to the template strand. So you might not be able to understand this here. Let me just draw it again so you will understand this. So here we have these two templates. You know about these templates. So here we have this this is the finger region, and so the template is in the red color, so you can see it is folded 90-degree. So the significance of 90-degree fold, I'll tell you. So these are the base pairs. I'll just draw this much, and then this is the primer. So this is the primer. These are the new formed base pairs. So here there is no base pair. So this one, this base pair on the template strand is the one for which we have to search the complementary base pair. And this one is the base pair which is next to this base pair, right? This is just like that. So there is an angle of 90 degree, and this angle is maintained so that the enzyme will not have any confusion that whether to search for this base pair or to this base pair, because it is 90 degree away, it is directly away from the site of catalysis. This will decrease the confusion, and whatever the DNTP, what the DNTP which is complementary to this strand, that DNTP come up and attach with this without any confusion, because this is out of sight. So that's why the 90 degree this 90 degree angle between these two base pair which are next to each other on the template stand is important, so that the confusion can be decreased. This is the another function of the finger domain. Next, there is thumb domain. So there is no direct function of thumb domain in the catalysis, but it is only involved in stabilizing. So this is it again has two function. One is to maintain the position of primer template junction relative to the active site. So this is the primer, and this is the template. So in order to maintain this position, and the another thing is it ensure the strong association between DNA polymerase and these substrates. So these two functions are there for the thumb region. Then I'll quickly explain, like I'll sum up all these three regions. So we'll make three DNA polymerases, and you know already, like we denote red is for like this, red is for the template, and then green is for the green, so green is for the primer. So here what happen is that first we have to see like which first we have to find the DNTP. So once the DNTP is complementary to the base pair which is present on the template, this finger region will come like this, and once this finger region is like this, the reaction happens, and once the reaction happens, this template strand will move one base pair ahead, and because of the movement, another base pair which another base pair will show up, and this position will open, and then again we have to find another DNTP for this position. So because of this palm region and because of this finger region, these two regions are important for the catalysis, and the thumb region is important for the stability of the substrate and the stability of the template and primer strand on the enzyme. So now we'll discuss the exonuclease activity also, because that is also important. You might knowing that this has a 3 dash to 5 dash exonuclease activity, but today I'll show you like how exactly it knows that whether a wrong base pair is there in the strand, and how exactly that exonuclease activity take place. So here as you can see that this is mispaired last base pair. So let's say that this one base pair is mispaired and it attaches with the primer strand. So what will happen is that because of this mispaired base pair, the hydrogen bond will not form. So you might be knowing that here I have discussed that this palm region will form the hydrogen bond with this strand, the newly formed strand. And once you are adding this let's say that you are adding, and by mistake you have added something which is not complementary. Let's say you have added this region. So this is something which is not complementary to the base pair here. So what will happen is that the hydrogen bond will not form, and the affinity increases for the exonuclease active site. So there is a site which is like an exonuclease active site. So tenfold increase in affinity happens. Affinity for exonuclease active site will increases, and because of this increase in the affinity of this strand with the exonuclease site, it will go and attach to the exonuclease site. And once it is attached with the exonuclease site, the exonuclease will remove that base pair which was there, which was not complementary to the base pair on the template region. And after the removal of the base pair, it will again go back here, and again it will kind of like it gives again a chance for the a chance to the enzyme to correct its mistake. So that's how that's the mechanism: decrease in the hydrogen bond interaction with the palm region, and then increase in the interaction with the exonuclease region, which will remove that base pair which is not complementary, and then again it goes back to the back for the correction of this mistake. But if this mechanism happens in the cell, then if the exonuclease activity is present, let's say a cell is there where exonuclease activity is not present, so it's the chance of mistake is 1 mistake in 10 to the power 5 base pairs. But if let's say it's not present, so if it is present, let's say the exonuclease activity is present, then the chance is 1 to the power 1 in 10 to the power 7 base pairs. So the chances has decreased significantly. But when the scientist has observed exactly what is happening, so they have observed that in our body there is 1 mistake in 10 to the power 10 base pair. So it is way more than this also. So which means that this exonuclease activity is not enough. There is something else which is also working with this exonuclease activity to even decrease the chances of mutation or chances of error while replication, and that we will discuss later, how these chances are even decreased after 10 to the power 7 base pair. So after that, the another thing which is there in this DNA polymerase, the another good thing about DNA polymerase is that DNA polymerase is a processive enzyme. So it's a processive enzyme. What does that processive enzyme mean? That once it is attached, so this is the primer, this is the template, and this is the primer, and this is the polymerase, let's say. So once it is attached with this template primer, it will lead to formation of this whole strand. It will not be like it forms, it add 1 base pair and then it goes away, and then again one another enzyme come and then it bind, and then it add 1 base pair and then it goes away. It's not like that. So after it binds, it can add up to like thousands of nucleotides in 1 second. So yeah, so this is the processive nature of the enzyme. And in next video we'll discuss the replication. So in how exactly the replication is happening. And so I hope you understood concepts in this video. If you're not understanding anything, please let me know in the comments. And yeah, thank you.